Assalamu alaikum. Our lecture is Extracellular Matrix Roles in Tissue Regeneration. The extracellular matrix is an extensive molecule network composed of three major components: protein, glucosaminoGlycan, and glycoconjugate. Extracellular components, as well as adhesive receptors, interact with each other, forming a complex network into which cells reside in all tissue and organ. In biology, the extracellular matrix is a three-dimensional network of extracellular macromolecules such as collagen, enzyme, and glycoprotein that provide structural and bio. The extracellular matrix is a structural support network made up of a diverse protein, sugars, and other components. It influences a wide number of cellular processes, including migration, wound healing, and differentiation, all of which is of particular interest to researchers in the field of tissue engineering. The extracellular matrix is the non-cellular component present within all tissue and organs and provide not only essential physical scaffolding for the cellular constituents, but also initiate crucial biochemical and biomechanical cues that are required for tissue morphogenesis, differentiation, and hemostasis. An essential part of holding capacity of tissue is the extracellular area. The extracellular region is primarily occupied by a complicated network of macromolecules constituted called as extracellular matrix. The composition of extracellular matrix is varied depending on the species and also developing or ground molecules. Commonly, the extracellular matrix is composed of three major classes of biomolecules, which are glycosaminoGlycan linked to a protein known as a Protobliton, and also fibrous protein including collagen, elastin, Fibronectin, Fibronectin, and laminin. This slide shows the structure of the extracellular matrix. This is the extracellular space contain the extracellular structure, which is the integrin, fibronectin, proteoglycan, collagen, laminin, and this is the basal plasma membrane of the cells, and this is the cytosols or cytoplasm, which contain the cytoskeleton inside the cells, and this is the attachment to the cytoskeleton by this is the integrin. Also, this picture shows the extracellular matrix of hyaline cartilage. This composed of protoglycan aggregate, where also collagen fibrils and large diameter of collagen fibril. In addition, a connective tissue is also composed of matrix of extracellular matrix. One of the essential components of connective tissue is fibroblast and the ground substance. The ground substance is a mixing complex between glucosaminoGlycan, protoglycan, and glycoprotein, mainly laminin and fibronectin. In most connective tissue, the matrix constituents are secreted by fibroblasts, but in several certain specialized types of connective tissue, like cartilage and bone, these components of extracellular matrix are secreted by chondroblast cells and osteoblasts. This picture shows the cells present in the connective tissue. This is the ground substance, the blood vessels, and this is the cells. This is also elastic fiber, collagen fiber, reticular fibers, and also this is the mesenchymal cells, fat cells, fibroblast cells, and etc. Role and importance of extracellular matrix. It regulates a cell dynamic behavior. In addition, it sequestrates a wide range of cellular growth factors and acts as a local depot for them. Changes in the physiological condition can trigger protease activities that cause local release of such depot. This allows the rapid and local growth factor mediated activation of cellular functions. Formation of extracellular matrix is essential for processes like growth, wound healing, and fibrosis. An understanding of extracellular matrix structure and composition also help in comprehending the complex dynamic of tumor invasion and metastasis in cancer biology, as metastasis often involve the destruction of extracellular matrix by enzymes such as serine and threonine proteases and matrix metalloproteinases. In general, all the cells need to attach to the extracellular matrix to grow and multiply. So the extracellular matrix provides support and encourage for the shape of the cells, regulate and determine cells dynamic and behavior, including cell survival, proliferation, polarity, cell differentiation, adhesion, and cell migration. Moreover, the extracellular matrix also gives the mechanical support for tissue and is involved in the growth mechanism, regenerative and healing process. Molecular components of extracellular matrix include protoglycan polysaccharide as heparin sulfate, chondroitin sulfate, and keratin sulfate, and non-proteoglycan polysaccharide like hyaluronic acid, also fibers like collagen and elastin, and adhesive glycoprotein like laminin fibronectin. Type of molecules in the extracellular matrix: structural protein for strength flexibility like collagen and elastin, protoglycan for matrix formation, and adhesive glycoprotein stick cells to matrix like fibronectin and laminins. This is the extracellular matrix with its component. This is the fibronectin, collagen, laminin, protoglycan, and this is the integrin, and this is the basal lamina, and this is the cytoplasm of the cells. The first component of extracellular matrix is glycosaminoglycan, or GAGs, are unbranched chain of polysaccharide, are composed of repeating disaccharide units and are heterogeneous group in negatively charged polysaccharide chain that are covalently linked to protein to form protoglycan molecules. The name of GAGs is because in this polysaccharide, one of the two sugars in the repetitive disaccharide is always as amino sugar, such as N-acetyl glucosamine or N-acetyl galactosamine. The second sugar of the GAGs usually is the uronic acid like glucouronic or iduronate. GAG molecules are negatively charged, as we said before, because there are sulfate or carboxyl group in the most of the sugar. A. This is the structure of a protoglycan monomer. And B. Example of individual glucosaminoglycan. This is the glucosaminoglycan chain, and this is the core protein. And C. The morphological of the protoglycan monomers. The five main groups of GAGs or glucosaminoglycan are differentiated based on the sugar type, including first, hyaluronan or hyaluronic acid, second, chondroitin sulfate, third, determined sulfate, 4 heparin sulfate, and 5 keratin sulfate. Hyaluronan is the simplest glucose aminoglycan and does not contain sulfate sugar. All disaccharide units are the same, and the chain length is extensively bigger. Thousands of sugar monomer. The hyaluronan is not connected conveniently to some core protein. Protoglycan are composed of glucose aminoglycan chain that are covalently linked to the core protein and considered to have a significant role in chemical signaling among the cells. This is the structures of protoglycan. This is the structure of protoglycan. This is the hyaluronic acid of hyaluronan, and this one is shown here. This core protein, and this is chondroitin sulfate, and this is a small one. Another picture of a protoglycan. This is the electronic pictures shown here. This is the hyaluronate or hyaluronic acid, and this is the chondroitin sulfate which attached to the core protein, and this is the small one represent the keratin sulfate, and this is the linker protein with hyaluronic acid. Protoglycan function: first, trap water; second, resistant to compression; third, return to original shape, occupy space, link to collagen fibers, and form the network. Second component of extracellular matrix is the collagen, which is the most abundant protein in the extracellular matrix. In fact, collagen is the most abundant protein in the human body and accounts for 90% of bone matrix protein content. Collagen is exocytosed in a precursor form, procollagen, which is then cleaved by procollagen proteases to allow extracellular assembly. Collagen, which constitute the primary structural element of the extracellular matrix, provide tensile strength, regulate cell adhesion, support chemotaxis and migration, and direct tissue development. Recently, there have been already described 28 types of collagen. The main type of collagen found in the connective tissue are type 1, 2, 3, 4, 5, and 11. Collagen polypeptide chain are synthesized on the membrane-bound ribosomes and feed into the lumen of the endoplasmic reticulum as large precursors called the pro-alpha chain. Each pro-alpha chain then joins the other two to form the hydrogen bond triple strand hydrogen molecule known as the procollagen. After secretion, the fibrillar procollagen molecule divides to become collagen molecule, which converge into fibril. This is the collagen fibers. This is a triple helix of the collagen fibers, and this is the electron microscope picture of the collagen fibers. This is the electron microscopical picture of collagen fibers, and this is the collagen fibers consist of collagen fibrils, and this is collagen fibrils composed of collagen molecules triple helix, which contain a 3-alpha chain. And this is also the formation of the collagen by a precursor alpha chain. 3 chains assembled together to form triple helix with loose end, and these procollagen peptidase form the collagen molecules, and this is assembly of collagen and this formation of collagen fiber. Types of collagen can be divided into several families according to the types of structure they form. as fibrillar, like type 1, 2, 3, 5, and 11, short chain types, and basement membrane types, and other types. Second type of fibers present in the extracellular matrix is the elastin. In contrast to collagen, give elasticity to tissue, allowing them to strain when needed and then return to their original state. Elastin are synthesized by fibroblasts, smooth muscle cells. Elastin are highly insoluble. The structures of elastin composed of protein, composed of amino acid glycine, valine, alanine, and proline, cross-linked tropoelastin monomers, first secreted as soluble precursor tropoelastin, assembly and cross-linking of tropoelastin monomers form insoluble elastin matrix into functional fibers, lysine residue in the cross-linking domain of secreted tropoelastin rapidly cross-linked both inter- and intramolecularly by lysyl oxidase. Hydrophobic segment elastin properties, alpha helical segment alanine and lysine rich, form cross-link between adjacent molecules. This picture shows the elastin structure. This is the elastin during the relaxation, and this is during the stretching. Each of this is represent the single elastin molecule which cross-link with each other by this cross-link. Elastin function, structural integrity and function of tissues, requiring a reversible extensibility or deformability, high level in tissue that require elasticity like in lung, skin, major blood vessels. Another molecules in the extracellular matrix is the adhesive glycoprotein, like fibronectin, laminin, and integrin. Fibronectin is an extracellular matrix that makes cell adhere to the matrix, is considered as a large glycoprotein found in all vertebrates, usually exists as a dimer composed of two nearly identical 250 kDa subunit linked covalently near the C-terminal by a pair of disulfide bonds at one end side. Fibronectin is a ligand membrane of the integrin receptor family. Integrin are structurally and functionally related to the cell surface as heterodimeric receptors that link the extracellular matrix with the index. The primary type of fibronectin is known as type 3, fibronectin replica, or called cylindral, which bind to the integrin. This model has a length of about 90 amino acids. Fibronectins appear in the soluble and fibrillar form. There are two other fibronectin isoforms, which are fibronectin type 1, hexagon, and fibronectin type 2, square. Fibronectin is not only crucial for attaching cells to the matrix, but also to guide cell migration in vertebrate embryo. Fibronectin has many functions which allow it to interact with many extracellular substances such as collagen, fibrin, heparin, and with a specific membrane receptor in the responsive cells. Fibronectin can be divided into two types according to the solubility. Soluble plasma fibronectin called insoluble globulin is a major protein component of blood plasma, 300 microgram per mol and is produced in the liver by hepatocyte cells. The second type is insoluble cellular fibronectin. Is a major component of the extracellular matrix. It is secreted by various cells, primarily fibroblasts, as a soluble protein dimer and is then assembled into an insoluble matrix in a complex cell-mediated process. Fibronectin plays a major role in the cell adhesion growth, migration, differentiation, and is important for the process such as wound healing, embryonic development. It is bound to many other host and non-host molecules. For example, it has been shown to interact with protein such as fibrin, tenascin, tumor necrosis factor alpha, and bone morphogenic protein 1. Role of fibronectin, as we said, it anchors cell to the extracellular matrix, maintains cell shape, cell movement, and blood clotting. Another adhesive molecule is the laminin. Are proteins found in the basal lamina of the virtually all animals. Rather than forming collagen-like fibers, laminin form network of web-like structure that resist tensile force in the basal lamina. The laminins are an important and biologically active part of the basal lamina, influencing cell differentiation, migration, adhesion, as well as phenotype and survival. Laminin found in basal lamina, special extracellular matrix and epithelial cells, separate them from the connective tissue. Role of basal lamina: support permeability barrier, contain type 4 collagen, protoglycans, and laminin. This is the extracellular matrix component. This is the laminin, as we see here. This is also the laminin. The structure of laminin is made up of 3 separate parts called the alpha, beta, and gamma chain that give it a total of 6 ends, which account for a lot of its flexibility in connecting up various kinds of molecules. Laminin is vital to making sure overall body structure holds together. This is the structures of laminin. This is in the electron microscope. Laminin networks form independent network and are associated with type 4 collagen network via intacin and perlecan. They also bind to cell membrane through integrin receptor and other plasma membrane molecules such as the dextroglycan glycoprotein complex and Lutheran blood group glycoprotein. Function of laminin for cell adhesion, migration pathways, stimulates growth of axons, development and regeneration, differentiation, basal lamina, most abundant linking glycoprotein. Tissue regeneration. Extracellular matrix is the primary factor required in the process of forming a new network and tissue. Along with the development found, many different factors can trigger the growth of extracellular matrix or used to create a synthetic extracellular matrix. Currently, extracellular matrix is involved in various mechanisms such as wound healing, with or without the involvement of mesenchymal conditioned media, and neuronal regeneration capability associated with pathological and or neurodegeneration disease like Alzheimer. In terms of injury repair and tissue engineering, extracellular matrix serves 2 main purposes. First, it prevents the immune system from triggering from the injury and responding with the inflammation and scar tissue. Next, it facilitates the surrounding cells to repair the tissue instead of forming scar tissue. The process of wound healing is strongly influenced by the role of migration, proliferation of fibroblasts in the injury site. Indeed, fibroblast is one part of extracellular matrix. The proliferation of fibroblasts determine the outcome of wound healing. So these cells will produce collagen that will link to the wound, and fibroblasts will also affect the process of reepithelialization that will close the wound surface. Fibroblasts will produce type 3 collagen during proliferation and facilitate wound closure. During proliferating stage, fibroblast proliferate activity is higher due to the presence of transforming growth factor stimulated fibroblasts to the secrete beta fibroblast growth factor. The higher number of fibroblasts also induce increasing of collagen synthesis. Collagen fiber is the major protein secreted by these cells composed of extracellular matrix to replace wound tissue strength and function. Also, collagen fibers deposition was significant on 8 to 10 days after injury. The number of fibroblasts increased significantly in correlation with the presence of an abundance of beta fibroblast growth factor on the day 8 to 10 after wounding. Thank you for listening.